The overall goal of this procedure is to map the genome-wide replication timing program of a cell of choice.
This method can help answer key questions in the replication field, such as how the genomic replication program changes in health and disease.
The main advantage of this technique is that it is a fast and simple way to map replication timing in various cells and conditions.
Demonstrating the FACS procedure will be done Lehmann, a FACS expert in the Faculty of Medicine.
This procedure usually starts with at least 1 to 2 times 10 to the 6th fast-growing unsynchronized cells. For adherent cells, aspirate and wash each plate with 3 milliliters of PBS without calcium or magnesium. Discard the PBS and add 1 milliliter of commercial trypsin EDTA to each plate. Incubate the cells for 5 minutes at 37 degrees Celsius until they detach. Add 3 milliliters of culture media to neutralize the trypsin and collect the cells in a 15 milliliter conical tube or a 5 milliliter polystyrene tube. Keep the cells on ice. To begin this procedure, centrifuge the cells at 300 times G for 5 minutes at 4 degrees Celsius. Aspirate and wash the cells with 1 milliliter of cold PBS. Then spin down the cells.